Good day everyone, I'm Anime Dismaya and I'm going to report the gene regulation in prokaryotes and eukaryotes. But before that, I'm going to have some background of genomes, proteomics, and the genetic code. Let's have first the genomes. Genome is an organism's complete set of genetic instructions. Each genome contains all of the information needed to build that organism and allow it to grow and to develop. Next is that the instruction in our genome are made up of DNA. Within DNA is a unique chemical code that guides our growth, development, and health. This code is determined by the order of the four nucleotide bases that make up the DNA, the adenine, cytosine, guanine, and thymine, ACG and T for short. So DNA has a twisted structure in the shape of double helix. Single strands of DNA are coiled up into structures called chromosomes. Next, our chromosomes are located in the nucleus within each cell. Within our chromosomes, sections of DNA are read together to form the genes. Genes control different characteristics such as eye color and height. All living things have a unique genome. The human genome is made up of 3.2 billion bases of DNA, but other organisms have different genome sizes. Next to that, this is the diagram of the genome. So, the genome inside the cell, the chromosome, and the DNA, and these are the genes. Next, the proteomics. The term proteome refers to the entire complement of proteins, including the modifications made up to a particular set of proteins produced by an organism or a cellular system. This will vary with time and distinct requirements, such as stresses that a cell or organism undergoes. Next to that, the term proteomics refers to a large-scale, comprehensive study of a specific proteome, including information on protein abundances, so their variation and modifications, along with their interacting partners and networks in order to understand cellular processes. So this diagram, this one represents chromosome are in single pieces of DNA that contains many genes. In the second diagram, DNA is a molecule that carries genetic information. So genes contain instructions for how to make proteins. And then on the third diagram, proteins act alone or in group to perform many functions. So, what are the unique challenges for proteomics compared to genomics? So let's answer the question. One gene can encode more than one protein, so even up to thousands. The human genome contains about 21,000 protein encoding genes, but the total number of proteins in human cells is estimated to be between 250,000 to 1 million genomes. Proteins are dynamic. So proteins are continually undergoing changes. Example, binding to the cell membrane, partnering with the other proteins to perform complexes, or undergoing synthesis and degradation. The genome, on the other hand, is relatively static. So proteins are co- and post-translational modified. So as a result, the types of proteins are measured can be vary considerably from one person to another under different environmental conditions, or even within the same person at different ages or states of health. Additionally, certain modifications can regulate the dynamic of proteins. So proteins exist in a wide range of concentrations in the body. So for example, the concentration of the protein albumin in blood is more than 1 billion times greater than that of interleukin 6, making it extremely difficult to detect the low abundance proteins in a complex biological matrix such as blood. Scientists believe that the most important proteins for cancer may be those found in the lowest concentrations. So, the genetic code. The genome of an organism is inscribed in DNA or in some viruses RNA. The portion of the genome that codes for protein or an RNA is referred to as a gene. Those genes that code for proteins are composed of trinucleotide units called codons. So each coding for a single amino acid. Each nucleotide subunit consists of a phosphate, deoxyribose sugar, and one of the 4 nitrogenous nucleotide bases. The purine bases, adenine and guanine, are larger and consist of 2 aromatic rings. So this diagram represents the ribonucleic acid. So it has codons. And again, 1 codon is equal to 1 amino acid. And this represents the DNA structure. So it's sugar phosphate backbone, the red line, and the nitrogenous bases, its major groove, and then the minor groove. So the red ones represent the base pairing between bases on the same strand can introduce a bend or hairpin in the molecule. The green line shown in green, base pairing can also occur between 2 different strands of DNA. So A to U, G to C, U to A. So the pyrimidine bases, cytosine and thymine, are smaller and consist of only 1 aromatic ring. In the double helix configuration, 2 strands of DNA are joined to each other by hydrogen bonds in an arrangement known as base pairing. These bonds almost always form between an adenine base on one strand and a thymine on other strand, and between a cytosine base on one strand and a guanine base on the other. This means that the number of A and T, or the adenine and thymine residues, will be the same in a given double helix as will the number of guanine and cytosine residues. In RNA, thymine is replaced by uracil, and the deoxyribose is substituted by ribose in RNA. So these are the nitrogenous bases and their structures. So the adenine, guanine, thymine, cytosine, and then the uracil. So this diagram shows the genetic code, and these are the blue color are the amino acids. There are various forms of gene regulation that is mechanism for controlling which genes get expressed and at what levels. But let's have the regulation of gene action in prokaryotes. Francois Jacob and Jacques Monod in 1961, they are the 2 French geneticists discovered while studying bacteria that the enzymes synthesized by them can be placed into two categories. The first one, those bacteria that are synthesized all the time and occur in relatively constant concentrations, such as enzymes of glycolysis. The second one, those bacteria that are synthesized only after a specific stimulation. The first type was named constitutively synthesized, and the latter the inducible enzymes. So, analyzing a variety of Escherichia coli that were defective for the induction of the lactose-utilizing enzymes, Jacob and Monod hit upon the possible molecular mechanism that controls the repression and derepression of a set of genes. The E. coli requires a set of 3 genes to be able to metabolize lactose. So when a little lactose is added to a glucose-free growth medium, it is seen that these 3 lactose-utilizing genes, or the LAC genes, named as LAC-Z, LAC-Y, and LAC-A, are synthesized simultaneously. Those 3 LACs, they are genes. Okay, next. So, the product of LAC-Z is the enzyme beta-galactosidase that catalyzes the conversion of lactose into galactose and glucose. So, these genes are not expressed in the absence of lactose. Jacob and Monod proposed the operand model to explain the genetic basis of induction and repression of lactose genes in prokaryotes. They were awarded Nobel Prize for this work in 1965. The operon. So operons are segments of genetic material, the DNA, that function as regulated unit that can be switched on or switched off. So, an operon consists of minimum 4 types of genes. Regulator, the operator, promoter, and structural. So, regulator gene is a gene which forms a biochemical for suppressing the activity of operator gene. Operator gene is a gene which receives the product of regulator gene. So it is the receiver of the product of the regulator gene, the operator gene. So it allows the functioning of the operon when it is not covered by the biochemical produced by a regulator gene. The functioning of operon is stopped when operator gene is covered. Next, promoter gene is the gene which provides point of attachment to RNA polymerase required for transcription of structural genes. Structural genes are genes which transcribe messenger RNA or mRNA for polypeptide synthesis. An operon may have 1 or more structural genes. Example, 3 in LAC operon, 5 in tryptophan operon, 9 in histidine operon. So the polypeptides may become component of structural proteins, enzymes, transport proteins, hormones, antibodies, etc. Some structural genes also form non-coding RNAs. The mechanism for regulation of protein synthesis utilizing operon model can be illustrated using 2 examples, the lactose and tryptophan in bacteria. This is the genetic map of the LAC operon. So in site A, the L represent as the regulator or repressor gene. The P serves as the promoter, and the O is the operator. The structural genes are the LAC-Z, LAC-Y, and then the LAC-A. In site B, in this site, the Escherichia coli operon is repressed. And in this site, inside B, the operon is induced. So, in this site, the L produces the RNA polymerase and 2 mRNA. So repressor or co-repressor, this figure is the repressor-co-repressor complex. And then the repressor binds to the operator and prevent transcription of Z, Y, and A genes. Hence, enzyme synthesis does not take place because it binds to the operator to prevent transcription to the structural genes. In this site, so as what I said a while ago, it is induced. So the structural genes produces beta-galactosidase, galactoside permase, galactoside acetylase, enzyme synthesis takes place. So there is no binding to the operator. And then the inducer-repressor, the inducer-repressor complex cannot bind to the operator. That's why the enzyme synthesis takes place. So, for more, inducible operon system or the induction of operon. Inducible operon system is regulated operon system in which the structural genes remain switched off unless and until an inducer is present in the medium. So it occurs in catabolic pathways. The lock operon of Escherichia coli is an inducible operon system which was discovered by Jacob and Monod in 1961. The lock operon of Escherichia coli, as what I've said, they are the three structural genes, the Z, Y, and A. In the induced operon, the structural genes transcribed a polycistronic messenger RNA which produces three enzymes. So they are the beta-galactosidase, galactoside permase, and galactoside acetylase. So they are the three enzymes. And as on my diagram before, I've shown to you these three enzymes. Beta-galactoside brings about hydrolysis of lactose or galactoside to form glucose and galactose. Next, galactoside permase is required for entry of lactose or galactoside into the bacterium. Galactoside acetylase is a acetylase which can transfer acetyl group to beta-galactoside. Again, the three enzymes which produced in the structural genes are beta-galactoside, galactoside permase, and galactoside acetylase. So the initiation codon of structural gene Z is TAG, corresponding to AUG of messenger RNA, and is located 10 base pairs away from the end of the operator gene. The substance whose addition induces the synthesis of enzyme is called inducer. Inducer is a chemical which attaches to repressor and changes the shape of operator binding site so that repressor no more remains attached to the operator. In the lock operon, allolactose is the actual inducer, while lactose is the apparent or visible inducer. Inducers, which induce enzyme synthesis without getting metabolized, are called gratuitous inducers. Like for example, the IPTG or isopropyl thiogalactoside. Regulator gene, it is a gene that produces a messenger RNA that synthesizes a biochemical repressor. Repressor is a small protein formed by a regulator gene which binds to operator gene and blocks structural enzymes, thus checking mRNA synthesis. The repressor of lac operon is a tetrameric protein having a molecular weight of 160,000. It is made up of 4 subunits, each having molecular weight of 40,000. The repressor protein has 2 sites, a head for attaching to operator gene and a groove for attachment of inducer. Promoter gene functions as a recognition point for RNA polymerase. RNA polymerase initially binds to this gene. It becomes functional only when it is able to pass over the operator gene and reach structural genes. Operator gene controls the expressibility of the operon. It is normally switched off due to binding of repressor over it. However, if the repressor is withdrawn by the inducer, the gene allows RNA polymerase to pass from promoter gene to structural gene. Next to that, in lac operon, the operator gene is small, 27 base pairs long. The gene is made up of palindromic or self-complementary sequences. That is the lac operon. If lactose is added, the repressor is rendered inactive so that it cannot attach an operator gene and synthesis of mRNA takes place. Transcription is under negative control when lac repressor is inactive by inducer. Transcription in lac operon is under positive control through cyclic AMP receptor protein, the CAFP. The catabolite gene activator protein or CJA protein or cyclic AMP receptor protein binds to the CJA site. So when CAP is attached to the binding site, the promoter becomes a stronger one. CAP only attaches to the binding site when bound with CAMP or CAMP. When glucose level is at high CAMP, it does not occur and so CAP does not bind and hence RNA polymerase do not bind, resulting in low transcription. So lac operon will not, however, remain operative indefinitely despite the presence of lactose in the external environment. Then it will stop its activity with the accumulation of glucose and galactose in the cell beyond the capacity of the bacterium for their metabolism. Next, the repressible operon system, the repression of operon, like for example, the tryptophan operon of Escherichia coli. So, irrepressible operon system is a regulated segment of genetic material which normally remains operational but can be switched off when its product is either not required or crosses a threshold value. This system is commonly found in anabolic pathways. Tryptophan operon of Escherichia coli is one such repressible operon system. So, tryptophan operon has 5 structural genes, the E, D, C, and B, A. The gene E and D encodes for enzyme antranilate synthetase, the gene C for glycerol phosphate synthetase, gene B for beta subunit of tryptophan synthesized, and A for alpha subunit of tryptophan synthesized. regulator gene or tryptophan R produces a biochemical, generally a proteinaceous substance called aporepressor. The aporepressor alone is unable to block the operator gene because of the absence of the binding head. Therefore, the operand system remains switched on. A complete repressor is formed only when a non-proteinaceous co-repressor joins the aporepressor. Co-repressor is a non-proteinaceous component or repressor, which is also an end product reaction catalyzed by enzymes produced through to the activity of structural genes. The co-repressor combines with aporepressor and forms repressor, which then blocks the operator gene to switch off the operon. So this is the tryptophan operon of the E. coli. So this is the regulator gene to mRNA and to the aporepressor. So the promoter, this is the RNA polymerase, the operator and the promoter, the structural genes of the tryptophan operon of E. coli, so the tryptophan E, tryptophan B, C, B, and A, which produces enzymes EDCBA to the core repressor. So that is the tryptophan operon of the E. coli. So the structural genes stop transcription, and the phenomenon is known as feedback repression. So, co-repressor of tryptophan operon is amino acid tryptophan. In tryptophan, the repressor gene is not adjacent to promoter but located in another part of E. coli genome. Promoter gene or the tryptophan E is the recognition as well as initiation point for RNA polymerase. RNA polymerase attaches to promoter gene. It can pass to structural genes provided the operator gene is in the functional state. Operator gene, the tryptophan O, lies in the passageway between the promoter and structural genes. Normally, it remains switched on so that RNA polymerase can pass over from promoter gene to structural gene and bring about transcription. The operator gene can be switched off when both aporepressor and corepressor join together to form a repressor. The repressor binds to operator gene to interrupt movement of RNA polymerase. In absence of tryptophan, the RNA polymerase binds to the operator site, and thus structural genes are transcribed. So these are the tryptophan genes. So the tryptophan P, which is the promoter gene, the tryptophan O, it is the operator gene. Next to that, the transcription of structural gene leads to the production of enzyme, the tryptophan synthetase, that synthesizes tryptophan. When tryptophan becomes available, the enzymes for synthesizing tryptophan are not needed. So the corepressor or the tryptophan repressor complex blocks transcription. One element of tryptophan operon is the leader sequence or the L that is immediately 5' of tryptophan E gene. This L sequence controls expression of the operon through a process called attenuation. So attenuation is the termination of the transcription prematurity at the leader region. So the tryptophan operon is a negative control. So, in summary, the two operand models, no, the two operand models described a while ago can be summarized here. So the inducible system and the repressible system. The inducible system, it is the active repressor plus operator and system of. Active repressor plus inducer equal to inactive repressor, the system is on. In repressible system, aporepressor and corepressor complex equal to the active repressor, the system is off. Then aporepressor equal to inactive repressor, the system is on. Regulation of gene action in eukaryotes. Operands are also functional in eukaryotes. Likewise, they are of two types, inducible and repressible. So, gene regulation of eukaryotes is slightly complex as compared to that of prokaryotes. So, different structural genes connected to a metabolic pathway do not often lie adjacent to one another. They are generally found well spaced on the same or different chromosomes. Each structural gene seems to have its own promoter gene. Eukaryotes possess sensor genes which pick up information of any change in the intracellular environment and the presence or absence of hormones, vitamins, metal ions, chemicals, pathogens, etc. Eukaryotes have integrator genes for coordinated function of structural genes present in different parts of genetic material. Eukaryotes seem to have specific genes that enhance or slow down the expression of certain genes. They are called enhancer genes and silencer genes, respectively. Eukaryote structural gene has two types of interspersed regions. They are the exons and introns. Exons are coding sequences of a gene which on transcription form parts of mRNA that code for different regions of protein. Introns are non-coding sequences of the gene which are also called spacer DNA or intervening sequences, so they do not form parts of RNA. The whole gene is transcribed as a heterogeneous nuclear RNA. The non-coding or intronic regions of RNA are removed by means of enzymes nucleases. So the exonic regions of RNA are joined together to produce a single chain RNA required for functioning as translational template. The phenomenon of removal of non-coding regions and fusion of coding parts of RNA is called splicing. So, splicing again, it is the phenomenon of removal of non-coding regions and fusion of coding parts of RNA. The freshly formed messenger RNA undergoes several changes at 5 and 3 ends. It receives a cap at the 5 end and a poly-A tail at the 3 end. The tailored mRNA is then transported across the nuclear envelope for translation through ribosomes and tRNAs. So this is the gene expression in eukaryotes. So in cytoplasm, this one is the nuclear pore, the nuclear envelope. So this is the site of the expression in eukaryotes. So these are the structural gene. So it has 5 end and 3 ends. The promoter, exon, the intron 1, the exon 2, the intron 2, exon 3, intron 3, exon 4. So the transcription and this is the splicing and processing, the mRNA, then outside the transport, and this is the translation. In this part, this is the polypeptide, the polyribosome or polysome. So that is the gene expression in the eukaryotes. Operon operator model. So it is also known as gene battery model. It was proposed by Britten and Davidson in 1961. There are 4 types of genes, namely sensor gene, integrator gene, receptor gene, and producer gene. The integrator gene is likely a regulator gene of prokaryotes, which synthesizes an activator RNA for activating the receptor site. The activity of integrator gene is controlled by the sensor gene. The producer gene analogous to structural gene of prokaryotes. So the receptor gene is a link between integrator and producer gene. So the receptor gene gets signal from integrator gene. As a result, it activates the producer gene. So these are the site of genes. So the S stands for the sensor gene, the L for the integrator gene, the R for the receptor gene, and the P for the producer gene. So from the integrator gene, it produces the activator RNA to the receptor gene for the producer gene, which produces a protein. So the importance of gene regulation. There are 2 types of gene action, constitutive and regulated. The constitutive gene action occurs in those systems which operate all the time and the cell cannot live without them. Example, the glycolysis. It does not require repression. Therefore, regulator and operator genes are not associated with it. In regulated gene action, all the genes required for multi-step reaction and can be switched on or off simultaneously. It is in regulated gene. The genes are switched on or off in response to particular chemicals, whether required for metabolism or are formed at the end of metabolic pathway. The gene regulation is required for growth, division, and differentiation of cells. It brings about morphogenesis. That's all. Thank you for watching. I hope you understand my report.